Imagine reaching into the microscopic world, not with mechanical fingers or metal probes, but with pure light. Imagine sculpting that light into invisible hands so precise, so delicate that they can grasp a single DNA strand, unfold a protein, or hold a virus particle mid-air without ever making contact. This is not a fantasy of science fiction. It is the emerging reality of plasmonic nanotweezers, a frontier where light becomes a tool for precision molecular manipulation, and where physics, nanotechnology, and biology converge at the scale of individual molecules. At its core, this revolution begins with plasmons, quantized oscillations of electrons on the surface of metallic nanostructures. When light of the right frequency strikes these nanostructures, it does not simply reflect or scatter. It ignites. It concentrates into electromagnetic hotspots, 1,000 times smaller than the wavelength of the light itself. Within these hotspots, optical forces are amplified to such a degree that they can trap and maneuver nanoscale matter in ways previously unimaginable. This is the domain of plasmonic nanotweezers, nanoscale light traps that overcome the classical diffraction limit, enabling direct manipulation of biomolecules, not in bulk but one at a time. DNA, proteins, exosomes, ribosomes, all can be isolated, twisted, stretched, or rotated in real time. We are no longer just observing biology. We are beginning to interact with the machinery of life one molecule at a time, using nothing but engineered light and carefully designed nanostructures. How is such precision possible? Traditional optical tweezers use tightly focused laser beams to trap micrometer-sized objects, relying on the gradient of light intensity. But at the nanoscale, the force is too weak, the trap too loose. Enter plasmonics. By coupling light into gold or silver nanoantennas, bow-tie gaps, or nanohole arrays, we shape the optical field into ultra-confined regions where gradient forces skyrocket. These forces can now manipulate particles smaller than 10 nanometers, roughly the size of a small protein. But this is more than mechanical manipulation. These hotspots also localize heat and enhance optical signals, allowing simultaneous spectroscopy and trapping. We can monitor conformational changes of single proteins as they fold or unfold in response to mechanical stress. We can watch enzymatic reactions in real time. We can measure binding affinities, reaction kinetics, and even force-induced biological transitions with femtonewton resolution. Every molecule becomes an experiment, a story waiting to be read. In this new paradigm, biophysics becomes interactive. Imagine tuning the trap stiffness in real time to mimic the dynamic environment inside a living cell, or applying periodic optical pulses to simulate mechanical vibration and observe the mechanical resilience of biomolecular structures, or coupling the plasmonic tweezers to feedback-controlled AI systems that learn how to manipulate molecules for optimal reactions. Light-guided molecular robotics, in essence. Of course, the challenges are profound. The same light that traps can also burn. Thermoplasmonic heating must be managed delicately, especially when working with fragile biological molecules. The design of nanostructures must be optimized for both field enhancement and biocompatibility. And the stability of the trapping field must endure the relentless jostling of Brownian motion. Yet, as nanofabrication, computational design, and optical control systems evolve, these challenges are being met, one photon at a time. What are the implications of this technology? In the short term, single-molecule studies will accelerate, enabling discoveries in structural biology, enzymology, and synthetic biology. But in the longer view, plasmonic tweezers may become part of integrated lab-on-chip systems for disease diagnostics, drug discovery, and personalized medicine. We may see nanoscale robotic platforms where light-controlled systems can sort, sequence, or even assemble molecules on demand. And perhaps most provocatively, by giving us the ability to apply precise forces to individual molecules, we are beginning to explore mechanobiology at the quantum edge, where quantum coherence, plasmo-exciton coupling, and near-field interactions begin to influence biological behavior in ways we are only beginning to suspect. In this realm, the tweezers are not tools, but questions made manifest in light. Each manipulation an experiment, each measurement a window into the rules of life at its smallest scale. We begin to ask, Can a photon fold a protein better than a chaperone? Can light uncover hidden mechanical codes in the structure of DNA? Can we build logic gates from molecules controlled by light with the same fidelity as silicon chips? As we advance, plasmonic nanotweezers become more than instruments. They are bridges between physics and life, enabling a dialogue between photons and proteins, between matter and energy, at the scale where nature writes its most intimate code. Plasmonic nanotweezers use metal nanostructures to convert incident light into highly confined electromagnetic fields called surface plasmons. These fields generate gradient forces strong enough to trap particles much smaller than the diffraction limit of conventional optical tweezers. By concentrating optical energy into volumes thousands of times smaller than a cubic wavelength, plasmonic traps achieve femtonewton scale forces at milliwatt laser powers. This enables stable manipulation of individual molecules and nanoparticles in fluidic environments. Example, a focused 1064 mm laser on a 310 mm gold nanoaperture stably traps a 50 mm polystyrene sphere with under 2 MW of input power. Analogy. It's like creating a microscopic whirlpool of light that holds a single grain of sand steady amid turbulent water. Plasmonic nanotweezers are engineered metallic nanostructures, such as bow-tie nanoantennas and double nanohole apertures, that localize light into nanoscale hotspots for contactless particle manipulation. They generate intense near-field gradients that pull and hold objects as small as proteins, DNA strands, or viruses without fluorescent labels. Unlike conventional tweezers, plasmonic traps operate at low optical powers and require only simple optics. Their compact footprint makes them ideal for integration into microfluidic devices. Example, bow-tie nanoantennas on a glass substrate trap individual E. coli cells for live imaging and growth studies. Analogy, it's like crafting an invisible pair of tweezers made of light that can pluck a single hair from a haystack without disturbing any surrounding strands. Localized surface plasmon resonances, LSPR, occur in discrete nanostructures, confining light to nanoscale volumes, ideal for trapping molecules, while surface plasmon polaritons, SEPPs, propagate along metal dielectric interfaces to transport microscale particles. LSPR traps provide sub-100 nm spatial resolution and directed transport under fluid flow. Both rely on enhanced near-field gradients but differ in confinement and application scale. Combining LSPR and SPP strategies expands functionality from single molecule trapping to high-throughput particle sorting. Example, gold nanopyramidal dimers use LSPR to capture individual DNA molecules in ~10 m gaps, while patterned gold stripes use SPPs to guide polystyrene beads along defined pathways. Analogy, LSPR is like a pinpoint spotlight attracting a moth, and SPP is like a conveyor belt carrying packages down a factory line. Self-induced backaction, SIBA, enhances plasmonic trapping stability by coupling particle motion to shifts in a nanocavity's resonance. When a trapped particle drifts, the cavity resonance shifts, reducing transmission and generating a restoring force that deepens the optical potential well. This feedback mechanism significantly lowers required laser power compared to passive plasmonic traps. SIBA enables stable trapping of sub-50 m particles under fluid flow with milliwatt-level illumination. Example, a 310 N gold aperture employs SIBA to hold 20 m nanoparticles at less than 10 MW of power, while fluid flows at 10 UL/min. Analogy, it's like a smart leash that automatically tightens when the pet wanders too far and loosens when it stays close. Key plasmonic architectures, double nanohole apertures, bow-tie nanoantennas, and fiber-tip nanoapertures maximize near-field enhancement and minimize trap volume for label-free single-molecule detection. Double nanoholes enable real-time monitoring of binding events via transmission fluctuations. Bow-tie antennas confine fields and nanogaps to trap DNA and proteins with sub-nanometer precision. Fiber-tip apertures extend trapping into 3 dimensions and enable in vivo application. Example, double nanohole tweezers detect binding between BSA and anti-BSA molecules by discrete jumps in optical transmission. Analogy. It's like selecting the perfect set of tweezers, fine tip for salt grains, or needle nose for tiny beads. Based on the object's size and shape, Plasmons have trapped and released individual biomolecules, including 2 nm peptides, 5 nm proteins, and 30 nm viruses, in aqueous solution without labels. Real-time monitoring of optical transmission reveals conformational changes and binding kinetics at millisecond resolution. These experiments demonstrate non-invasive, high-precision interrogation of molecular dynamics. Such capabilities open new avenues for studying fundamental biological processes at the single molecule level. Example: researchers captured a free 2 nm peptide in a plasmonic nanoaperture and monitored its folding/unfolding dynamics via transmission modulation. Analogy: it's like catching a butterfly mid-float and watching its wing beats slow down under a magnifying glass. Integrated with microfluidics, Plasmonic Nanotweezers enable label-free single-cell sorting, rapid pathogen detection, and multiplex immunoassays with femtomolar sensitivity. They dramatically reduce assay time and sample volume for point-of-care diagnostics. Real-time trapping and sensing on a single chip allow detection of disease biomarkers at clinically relevant concentrations. This technology promises portable, high-throughput diagnostic platforms for healthcare and environmental monitoring. Example: a multiplex LSPRM microfluidic chip quantified 6 serum cytokines in 40 minutes at pg/ml detection limits. Analogy: it's like a miniature laboratory on a chip that can pluck a single needle from a haystack of molecules and immediately identify it. Key limitations include photothermal heating that risks biomolecule denaturation, two-dimensional confinement of traps, and fabrication complexity of reproducible nanostructures. Excess heat can cause thermal drift and damage sensitive samples during prolonged trapping. Achieving stable three-dimensional manipulation remains challenging due to near-field confinement. Overcoming these hurdles is essential for translating plasmonic tweezers into routine clinical and biological workflows. Example: bowtie traps required surfactant additives to prevent protein denaturation from local heating during extended measurements. Analogy: it's like walking a tightrope in a sauna, balancing delicacy under intense heat without losing stability. Emerging strategies combine electrothermoplasmonics for three-dimensional transport, surface-enhanced Raman spectroscopy for simultaneous chemical fingerprinting, and fiber-optic plasmonic probes for in vivo sensing. Smartphone computational platforms and AI-guided trap optimization promise democratized nanoscale diagnostics. Next-generation devices aim for autonomous, high-throughput single-molecule sorting and analysis at point of care. These innovations could revolutionize precision medicine and synthetic biology. Example: a fiber-tip plasmonic tweezer captured proteins inside live tissue slices and streamed live optical readouts to an external detector. Analogy: it's like deploying a microscopic robotic surgeon that fishes out a single virus particle from living tissue and broadcasts a live video feed powered entirely by a beam of light.